Hello everybody, my name is Gabrielle and I'm currently a senior at Oglethorpe University. The title of my long talk today is titled Utilizing a Mouse Model System to Investigate the Development of Vaccines for Peanut Allergies. I myself have a peanut allergy, so I thought that this topic was very interesting and able to have a vaccine that really intrigued me. A little background on how I came to this conclusion. I was just searching on Galileo and I just saw food allergies, tree allergies, things like that. And so I was like, okay, let me look at peanut allergies. And then I thought, are there vaccines? What's the conversation about that? So that's why I picked this conversation. Because if we're able to develop a vaccine, then those who have severe allergic reactions, such as not being able to be in the same room as peanuts, or they could even end in death, then those women and children and adults won't have to worry about the consequences of eating peanuts, or even just being in the same room as a family member or friend eating peanuts. My outline for today, I will first give a little background on some statistics for peanut allergies. Then I'll talk about why we will develop a vaccine. Like, why is that important? And then I will introduce an experiment that did some research to prevent peanut allergic reactions. Then I have my take-home messages and some references at the end. About 0.6 to 4.6 children in Western countries experience peanut allergies. This can develop early on in life with toddlers. So those who are not exposed to peanuts at an early stage in life are more likely to develop allergic reactions to peanuts versus those who are exposed at an early age. Me myself, I was exposed at an early age, but I still developed the peanut vaccine. So the body works in mysterious ways. So also during my research, I saw that it's not only it can not only develop with oral administration of peanuts, but it can also be like a skin contact of peanut oil from ointments and creams that contain peanut oil. For example, I have this maize over here, which is a breast cream that women use for their breast area when breastfeeding, which then can go in the toddler's mouth and then develop the peanut allergy. Also, during my research, I saw that boiling peanuts can lower the amount of RARH1, which is the protein in peanuts that causes allergic reactions. So boiling it can boil it out, and then it can lower the amount of allergic reactions that people would have. However, this is still not a vaccine, and so the only way to fully prevent peanut allergies would be with a vaccine, so that we don't have to worry about having those allergic reactions that can be scary. Okay, so a little background. We have the paper that I was studying talks about neutrophil activating proteins, abbreviated as NAP. And then we have the T helper 2, abbreviated as TH2, and then T cells, which are abbreviated as TRIGs, as I might refer to later on. This was talked about in their prior research from the paper I did today, but a little background of it will help us understand what I'll be talking about today. So first, T helper cells produce antibodies. There are two types. You have TH1 and TH2. TH1 cells stimulate cellular immune response and stimulate B cells, which you can see here, to produce IgM, which are found. IgM is immunoglobulin M, which are found in the blood, and it is the first type of antibody made in response to an infection. TH1 also causes other immune system cells to destroy foreign substances. And it is the first line of defense against infections. Then you also have, within the TH1, you have IgG1, which is important for mediating antibody responses against viral pathogens. And then the one that we'll be talking about mostly today was the Th2, which stimulates humoral immune responses that promote B cell proliferation, which basically means it kills B cells, and induces antibody production. And then T cells are just those that come in and help fight the body from infection by attacking the specific foreign particles that it's meant to attack. So the NAP can suppress the T helper 2 cells by activating regulatory T cells. The Th2 cell mediated immune response is critically important in the development of peanut allergies, so that's why they were working with those. Administration of the NAP in mice was found to reduce the expression of immunoglobulin E, which are cells that release chemicals causing an allergic reaction by an overreaction of the immune system. Thereby, the NAP can shift the antigen-specific T cell response from a Th2 to a Th1 phenotype. And this is what inspired the authors to investigate the effectiveness of the NAP in suppressing peanut allergies. Okay, then we have Bacillus Subtilis, which are spores that are novel biological vectors that are non-pathogenic and have been previously used as a probiotic, and they also produce antibodies. And so a little background on that is that once they're passing through the gastrointestinal tract, most of the spores will survive and even colonize and reproduce and produce new spores in the gastrointestinal tract. So the recombinant spores showed in a previous study from the authors that they could increase fecal IgA levels, which is immunoglobulin A, which protects mucosal tissues from microbial invasion and maintain immune homeostasis with microbiota, which basically means they help regulate and make sure everything is working perfectly. And then the study also talks about chlorotoxin B, conjugated NAP, which is abbreviated CNAP, which I will talk more about later. So the paper that I was reviewing today is titled Oral Administration of Recombinant Bacillus Subtilis Spores Expressing Helicobacter pylori neutrophil activating proteins suppresses peanut allergy via upregulation of Tregs, which are T cells, by May et al., which the study was done in China. The goal of the paper I have here in quotation marks was to determine whether oral administration of recombinant NAP spores could inhibit allergic inflammation in peanut allergy-induced mice via the upregulation of Tregs. In other words, they were using a mouse model to see if it's possible to develop a peanut allergy vaccine by inducing the peanut allergies in mice. They obtained 5-week-old female mice from a Nanjing Biomedical Research Institute, which is in Nanjing, China, I'm pronouncing that right. And all the procedures were performed according to the ethics committee. So once the Bacillus subtilis spores were prepared, they had 42 mice that were randomly divided into 7 different groups. Each mouse was orally sensitized with 10 milligrams of peanut solution along with 20 micrograms of chlorotoxin and 16.5 microliters of vodka, which the vodka was just a source of food grade ethanol. This was done for 6 weeks, as you can see right here, from 0 to 5. And it was increased at week 6 and 8. The 10 milligrams of peanut solution was increased to 50 milligrams. And the peanut solution was specific IgA in feces was measured after each treatment. And here are the 7 treatments. So basically they were measuring the amount of immunoglobulin A in the feces after these treatments. After right here too. We have the sham PBS, which is a phosphate solution that basically just cleans the cells. And then you have the spores, the CTB spores, the NAP, you have the CTB plus NAP, which is a combination of the combined NAP, and then you have the CNAP plus, this right here, and then you have the naive, which was the control group. And to assess whether the mouse peanut sensitization model was successful, the blood was drawn before treatment was forced to detect the serum of the peanuts. So the recombinant spore treatment, these right here, were carried out between weeks 9 and 12. I also want to note that the NAP here promotes Th2 cell differentiation and enhances the body's immune response. So all these are supposed to give different immune response so they could test whichever one was a better vaccine. And where is it? The PBS, which, as I said earlier, it was used to wash the spleen to measure the amount of T cell production. The mice were fasted for 2 hours prior to the oral administration so they can make sure that they were only measuring the peanut solutions within the mice. And the oral administration of the peanuts were given 3 times a week. To assess whether the treatment the treatments were conferred peanut allergy protection, that was done by increasing the number of the T cells between the 4-week, between the 9-week and 12-week period. Here we have some results. On the x-axis, you can see the peanut solution in the IgA, which, as I think I said earlier, the IgA was to protect the mucosal tissues from microbial invasion and maintain immune homeostasis. This was measured in OD 450. That is the value. The OD value is a measure of how much of they had a stain, and so it's how much yellow color was produced in the stain. So the more color meant the more pathogen was produced in the sample, and that was done at 450 nanometers. So between week 1 and 4, the amount was increased. We can see here that the naive barely had any peanut allergy, as they were the control group. So it is increasing each week, as you can see, but mainly increasing. I could draw on here, hopefully. They pointed out that you can mainly see an increase between these three groups right here. Which are these last three right here. So these were the ones that were likely to produce the immunoglobulin A in order to combat the peanut allergy. So they had a higher amount of working as a vaccine for peanut allergies. Okay. So here we have on the y-axis, you can see the peanut allergy. I don't think you can see my pointer. Why is it not going? Okay, there we go. For IgE, and then we have IgG1 and IgG2. I couldn't really find a difference between the two, so I'm not always sure why we have this difference, but I did find that the IgG2A is related to the Th1 antibodies, so that's why we see the increase in that. And that's basically what they were trying to do. They were trying to change it from the Th2 to Th1 phenotype so that it can produce not as not as much as the allergic reactions, so preventing allergic reactions, basically. That's what I'm trying to say. So yes, you can see that these do decrease right here, but it increases right here again for these last three, as we saw earlier in the previous picture. And the authors said that these results basically suggest that spores can induce an increase in Th1 type antibodies. They also tested the symptoms of the mice. So after the peanut challenge, the mice were treated with the NAP spores or the CNAP spores, and they experienced mild allergic symptoms, elevated body temperatures, and a reduced histamine release. Right here, we're just going to focus on the symptoms, which are more important. So you can see what was happening. 0 represents no symptoms at all. 1 represents scratching and rubbing around the snot and head area. 2 represents puffiness around the eyes and snout and reduced activity and or decreased activity with the respiratory rate. 3 indicates wheezing and labored respiration, and 4 indicated no activity after prodding or tremors and convulsions, and then 5 represent death. I like to point out that right here you can see this one had the lowest amount of symptoms, so basically had 0, between 0 and about 2, whereas everything else had kind of high, including the death rates. This one still, the NAP looked good too. However, the CNAP combined, which was the combined NAP, the protein was sorry, was lower. So that's the most productive group. Okay. In conclusion, the results suggested that the oral administration of recombinant B. subtilis spores expressing the NAP or the CNAP confer local and specific immune responses. Recombinant spores could shift the antigen-specific T cells, which were those cells that are fighting off the allergic reactions, from a Th2 type to a Th1 phenotype and increases the production of the T cells, thereby conferring effective protection against peanut allergies. All in all, it kind of worked. The creating the vaccine works more so for the NAP and the CNAP, but more so for the CNAP. Take home messages. Peanut allergies. I just want everybody to know that peanut allergies is a very serious condition, once again. So this is why I was interested in this topic. And peanut allergies is basically just an immune over-response from those antibodies, which the over-response was related to the Th2. So that's why they were trying to change it to the Th1. And all in all, vaccines are possible. I feel like there's still more research that needs to be done, but this is like a good basis to develop a vaccine, looking at those immunoglobulins. And I really hope that in the future we can create a vaccine so that people don't have to worry about peanut allergies, because I myself have been scared a couple of times eating the wrong things that you may not visibly be seen as having peanuts in them. So it's very scary. And here are my references. Thank you all for listening, and I hope you found this topic as interesting as I did.